Hello and welcome back. So today we're going to talk about acquired immunity. Last lecture we talked about innate immunity, and between innate immunity and acquired immunity, these are ways that we fight those invaders trying to get into our body and also cancer cells. So this is also known as your adaptive immune system. So we can adapt to many different kinds of foreign agents. You can go to another country and be immune to some things that you wouldn't expect to be because you do have an immune system that can actively stalk and eliminate very, very specific kinds of pathogens, not generically like the innate immune system does. The thing about the acquired immune system is that it is very, very, very specific. So you have cells that specifically target individual kinds or strains of bacteria or viruses or other kinds of organisms. So it does take time after you are first exposed to an antigen. An antigen is anything that's a foreign agent to the human body that shouldn't be in the human body. So when the acquired immune system cells first see this thing, it does take some time for them to respond to it. When it does respond to it, though, it's going to be systemic. That means it'll be found all throughout the body and not isolated just in your skin or one portion of your body. And once we're exposed to an antigen, our acquired immune system allows us to have memory of that. So if you've ever been exposed to the flu virus, you get very ill the first time, and then next time you're exposed to that same specific strain of virus, you won't actually get sick from it, or at least the sickness won't be that significant. Just a little flow chart showing you some of the different lymphocytes that we're going to talk about in regards to the acquired immune system. So we're going to talk today about cytotoxic T cells, helper T cells, regulatory T cells, memory cells. All of these are T cells, which are a subdivision of a lymphocyte. Okay, so we're not going to talk about natural killer cells because we've already talked about those, although it's considered to be a lymphocyte. So cytotoxic T cells are going to be involved in attacking very specific virally infected cells or cells that are infected with some kind of foreign agent. Helper T cells are going to aid or stimulate the activation of both T cells and B cells. We are going to have ways of regulating this acquired immunity through regulatory T cells, and these help to moderate T cell and B cell activity. And then anytime a T cell or a B cell is activated, it's going to form a subclass of cells called memory cells, which respond to that specific kind of antigen next time around that you're exposed to it. The second subclass of lymphocytes is going to be the B cells, and we'll talk about these as well. B cells, when they're exposed to a specific kind of antigen, are going to form these cells called plasma cells. So they'll just differentiate into plasma cells, and these plasma cells will secrete a very, very interesting protein called an antibody that will attack any kind of bacterial toxin, foreign agent, bacteria, or virus, but very specifically. So we do have two types of acquired immunity. These have evolved to help us to fight very specific antigens and things that are dangerous in our local environment. And the first one is called humoral immunity. Humoral immunity is going to provide immunity by producing antibodies. These are little disease-fighting proteins that your plasma cells, which are the B cells that that have been activated, are going to make. So this humoral immunity is going to be made by B lymphocytes. These antibodies made by the B lymphocytes are going to enter into body fluids. They can enter into interstitial fluid, breast milk, and also semen, plasma of your blood. So these antibodies can basically get anywhere they need to go. We're then going to also talk about cell-mediated immunity. This is another type of acquired immunity. And this is immunity that's provided by your T cells or T lymphocytes. And these are going to basically go after virally infected cells, cells that may have something inside of them that needs to be destroyed. Just a little graphic showing you our adaptive immunity. So our adaptive immunity is triggered by antigen presentation. So we have to be, there has to be some kind of a virus or a bacterium or a toxin or a venom, something that's very specific that is entered into our body. And so when these particular things come in, they may activate cell-mediated immunity, which is going to be, the division that's done by the T lymphocytes. And these T lymphocytes are going to directly attack and kill virally infected cells. And then also we may utilize the branch of the antibody-mediated immunity, or humoral immunity. This is done by the B cells that become activated. When B cells become activated, they produce antibodies. Antibodies are going to attack any kind of a toxin, a virus, or a bacterium. And both the physical attack and the chemical attack on these organisms are going to destroy them, hopefully, or neutralize them in some kind of way. All right. Well, with this acquired immunity, we have several problems that need to be overcome. First of all, how do these chemicals and cells not destroy the body? This is the big, big issue. So to understand how these cells and antibodies don't destroy the body, we have to understand the concept of self-antigens. So every cell in the human body has these proteins on the surface called major histocompatibility complex proteins. Okay, and so every cell has MHC1, major histocompatibility complex 1, and this will be a marker that will help T cells and B cells understand what's going on inside of our body cells. Cells that are only involved in immune response are going to have what we call MHC2 complexes found on their surface of their cells. So these are going to be really important, as you'll see in just a second, in different kinds of immunity that we utilize. Okay, so we have self-antigens, things that we can recognize as being part of ourselves. So another thing, or another concept that helps us to understand how these immune system cells don't kill us, is called immunocompetence. Each lymphocyte has to be able to recognize and bind to only one specific antigen. Okay, this is called immunocompetence. And then lastly, we have self-tolerance. Self-tolerance is where each lymphocyte must be unresponsive to self-antigens. So anything that you produce in the body, these lymphocytes cannot respond to it. If they did respond to it, then you'd have an autoimmune reaction and there would be destruction of your body. So when we basically take and do a liver transplant or any kind of organ transplant, if the person that we get the organ from doesn't have the exact same genetic makeup that we have, doesn't express the same proteins, the lymphocytes in the donor person and the person that's the recipient of those organs, their lymphocytes will go and destroy those organs. Okay, so if we match up perfectly, then the lymphocytes won't recognize those organs as being foreign, and we won't have the rejection reaction that we see in organ transplants. Now, in this process of becoming competent and exhibiting self-tolerance, only about 2% of all lymphocytes that go to their places of maturation are going to make it through this process. So you can imagine that 98% of lymphocytes don't make it through this process and are destroyed. So this is a very selective process. You cannot produce lymphocytes that recognize the proteins that your body produces. They must not be able to respond to it. Okay, if they did, it basically causes autoimmune reaction. Now, these B cells and T cells are created in the bone marrow, so they come from that hemocytoblast. And T cells are going to travel in the bloodstream. They're going to go to the thymus, and that thymus is going to be a place where these T cells are presented with your self-antigens. They're going to be presented with real antigens, but really self-antigens is the big thing that's going to happen in the thymus. So in the thymus, if these T cells are presented with self-antigens and they respond, they're going to be eliminated. If they don't respond, then they'll make it through the maturation process and head out into the body and perhaps end up in a lymph node at some point in time. Remember, only 2% of the cells are going to pass that process. 98% of the cells don't pass that process. B cells are going to go directly to lymph nodes, and this is where they're going to be matured, and this is where they'll be presented with antigens and learn how to be B cells and react and do the job they need to do. So they'll go out to the lymph node in order to mature. All right, so let's talk about the division of acquired immunity called cell-mediated immunity. Cell-mediated immunity is going to be done by T cells. We have subsets of T cells. One of the subsets of T cells is called a helper T cell. And when a helper T cell is activated, it's going to stimulate B cells to produce antibodies. And we'll talk about that a little bit more specifically. Another lymphocyte within the cell-mediated immunity is called cytotoxic T cells. Cytotoxic T cells are going to be the cells that wander around. They're going to monitor the body's cells, and they're going to recognize and eliminate tumor cells. They're also going to eliminate virally infected cells, and they do this by the release of these proteins that are going to cut holes into those cells that are infected or that are tumor cells. Then there's some other means they use as well, but they use these little proteins that cut holes into infected cells and destroy those cells. And this is just indicating kind of how we use those major histocompatibility complexes that we talked about. So if we have an infected cell, there will be antigens that will be basically stuck on these major histocompatibility 1 complexes, and they'll be displayed on the surface of the cell. Well, cytotoxic T cells will come by, and they will use their T cell receptors in order to look at that antigen. And if it sees an antigen, it's going to actually target this particular cell for destruction. Okay, so it has a way to recognize something that's going on inside of the cell. Now, if we didn't have these major histocompatibility complexes, then we couldn't see inside the cell. A virally infected cell or a cell that was cancerous wouldn't be able to have a way to display those abnormal molecules, proteins, or things that shouldn't be inside the cell, and the T cell wouldn't have a way to recognize that. Okay, but we do have these major histocompatibility complexes. They have a way of displaying the antigen. The T cell receptor allows us to see that the cell is infected and then be able to respond to it. And that's how cytotoxic T cells are going to work. Helper T cells, on the other hand, are going to utilize the MH2, the MHC complex 2, and you can see that if a microbe comes into this cell here, maybe called an antigen presenting cell, then the microbe will be digested. These proteins from the microbe will be placed upon the MHC2 complexes, and it'll be displayed for the helper T cell receptor to see and recognize. And when this helper T cell recognizes that, it can actually send out chemicals that can begin to cause immune responses to occur. So let me show you how these things work. We'll do the MH1 class proteins first. So here we have some kind of organisms that have gotten into the bacterium. They have infected the bacterium, and so your body will respond to that internally and maybe digest some of those particular things. If you have a tumor cell, then the tumor will actually produce proteins that will be abnormal. So these abnormal peptides, they will be incorporated into an MH1, the major histocompatibility complex 1 protein, as they're being made in the endoplasmic reticulum. They'll be sent over to the Golgi apparatus. The Golgi apparatus will package those up in little vesicles and then send them to the cell membrane. So at the cell membrane, the MHC1 complex with that foreign protein on the surface sitting inside of it will be displayed so that a cytotoxic T cell can actually recognize that. So just to kind of show you the sequence of this, so here we have a viral or bacterial antigen that's on that MH1 complex, MHC1 complex. So we have an infected cell or a tumor cell, and we have a cytotoxic T cell that is recognizing that particular cell. If we look down here, a little bit more complexly, we have the T cell receptor from the cytotoxic T cell. It recognizes the antigen that is displayed by the class 1 MHC proteins. Now, these particular T cells are called CD8 because they have an extra little protein here that recognizes it as a CD8. We just call it the CD8 protein. So this cell will actually communicate to that it's infected not only through this, but it will produce other chemicals that will be a stimulating chemicals that will stimulate the T cell to basically go through and destroy it. Okay, and I'll show you how it destroys it in just one second. All right, so we have to recognize that a cell is infected or is a cell that's cancerous. So once we can recognize, we'll activate the T cells. These T cells will sometimes become clonal in that they'll clone themselves and actively go through cell division and make more of themselves. Some of the T cells, specific T cells to that particular antigen, they will become memory cells. And they won't respond now, but if you're ever exposed to the antigen in the future, those memory cells will activate very, very rapidly and expand themselves very rapidly so that you can fight an infection off very quickly. All right, so some of these activated T cells will seek out and they will join with virally infected cells, or whatever the cell is, a tumor cell, and they're going to cause the lysis or destruction of that cell. If we kind of look at this process more specifically down here, so we have the T cell receptor has recognized the MHC1 complex with the antigen in there. So we are going to have perforin release. These are proteins that are going to be like saw blades, and they're going to destroy the plasma membrane. And we also have lymphotoxins that are released that will cause the disruption of various cell metabolic processes. So these two things combined cause the lysis of the cell that's infected, thus hoping to stop the spread of a viral infection or to stop the spread of a tumor that might be growing. Okay, now let's look at how the MHC2 complexes are utilized. So here we have an antigen presenting cell. So this is an antigen presenting cell. It presents antigens to helper T cells. So you can see it's engulfing some kind of a bacterial agent. It's using lysosomes to digest that particular bacterial agent. So here we have a digestion event occurring. So the endoplasmic reticulum is producing MHC2 complexes. These will join with the lysosome, and then eventually they will travel to the plasma membrane and present, so that MHC2 complex will present the antigen to the helper T cells. So here we have the foreign agent inside of the antigen presenting cell. It's being presented to an inactive helper T cell. When that helper T cell though joins with the antigen presenting cell, there will be a stimulation that will occur of that helper T cell. That helper T cell will become activated. It will clonally expand itself, go through cell division. It will produce inactive memory cells that will go and kind of reside in different places in the human body, waiting to be presented by that antigen again. Now these activated helper T cells, these things are going to produce cytokines. Cytokines are very powerful hormones that are going to aid in stimulating both cell-mediated and antibody-mediated immunity. Okay. Now these cytokines are really, really super important. Now, if you ever hear about the flu epidemics that kill large numbers of people, we think that like for example the flu that occurred in 1918, that Spanish flu, it killed millions and millions of people. Like healthy people would get the flu virus like one day and be dead in a couple of days. And what we think that happens in some of these flu epidemics is that the virus comes in, it hyperactivates release of cytokines, and we get what they call a cytokine storm, which hyperactivates immunity and causes the death of a person. So So, it's a pretty important thing to kind of understand these cytokine storms, because when we have epidemics that kill large numbers of people, like bubonic plague and flu, you have to wonder how a person can be normal one day and then in a day or two be dead. And we think that our own immune system may be causing some of those mass mass mortality events that occur in epidemics. Okay, so just to remind you of what we just talked about. So if we use the class 1 MHC proteins, we are going to activate the cytotoxic T cells. So this has a way. This is a way that we can recognize virally infected cells, cells that are infected with bacteria, or cells that are tumors. And we will activate cytotoxic T cells to destroy those abnormal cells. We'll take and activate or make clones in memory cells, which will wait for the antigen to present itself again sometime in the future. And those cells will also produce regulatory cells so that the immune response isn't like too much and perhaps be destructive to the human body. Okay, so that's how we use the class 1 MHC proteins. Now, all of our cells have those on there and can present when there's something wrong inside of them. So you can't hide from your immune system. So class 2 MHC proteins are going to be utilized by helper T cells, and they're going to indicate the presence of pathogens, toxins, or foreign proteins. And we utilize antigen presenting cells. Those are the cells that are going to have class 2 MHC proteins on them. So these helper T cells, they're going to stimulate immune response amongst T cells and B cells, and they're going to produce memory cells as well to await the reappearance of that particular antigen so that it can respond very rapidly next time it sees that particular foreign agent. Memory T cells are really important because they provide a no-delay response next time you are exposed to an antigen that you've seen before. And so, when you get a vaccine, you are utilizing memory T cells and also memory B cells in order to have a no-delay response against an antigen that's in the vaccination. If you acquire, if you actually get an infection, then next time you're exposed to that agent, you may not ever even get sick and not even realize you've been exposed to it because of the memory cells. So memory cells are really, really important. Now you do have memory T cells and you have memory B cells. So we have both of those, and they both provide a no delay response. All right, let's talk about the humoral immune response. Humoral is kind of a weird word, but it means fluids. So it's the immune response that occurs in the fluids of the body. And this is going to be under the purview of B cells. So some of the B cells are going to become activated, and when they become activated, they become plasma cells. And these plasma cells will secrete these really interesting proteins called antibodies. Like T cells, some of the B cells will become memory cells, and therefore they'll have a no delay response to future encounters with foreign agents. All right, so in order for a B cell to be activated and to work, it has to basically be challenged. So there has to be antigen challenge. This is the first encounter that a B site that's never been challenged before will have to undergo in order to become activated. Now this challenge will be with a specific antigen. A specific antigen will activate only specific kinds of B cells that can recognize that specific kind of antigen. So this has a lot of specificity to it. It's very, very, very specific. All right, so we'll look at how this kind of works here. So here we have a naive or inactive B cell. You can see it has class 2 MHC proteins, and it's going to be presented with an antigen. So it does have very specific antibodies that can only join with very, very specific antigens. So if an antigen binds to that specific antibody, then antigen binding will actually stimulate the cell to become sensitized. And you can see it'll actually display the antigen on its class 2 MHC protein. Okay, so now we have a sensitized B cell. So a sensitized B cell will become activated once it joins with a helper T cell. So that helper T cell is going to stimulate with cytokines that B cell to become activated. So it'll actually mature or differentiate into a plasma cell once the helper T cell activates it. So we have to have sensitized B cells. They then have to be activated by helper T cells by that cytokine stimulation. Those hormones are going to leak across and activate that B cell. Once that B cell is activated, it's going to clone itself, and it's going to become many of the same. I mean, they're clones. They're genetically identical. So that B cell is going to become very, very, very quick to go through cell division, and then these activated B cells will differentiate into plasma cells. Plasma cells will be the active form of the B cell. These plasma cells are going to ramp up their production of endoplasmic reticulum and Golgi apparatus so that they can pump out humongous quantities of antibodies. Those antibodies will be in the fluids of your body, the blood plasma and the interstitial fluid or breast milk or semen. Those antibodies will be in that. Also, this activated B cell will form inactivated memory cells, and those will go and reside in all different places of your body waiting for that antigen to come around again so it can have a no delay response at producing these antibodies. Now this process you see here takes time. Okay, so sometimes you get sick and it takes a week or a week and a half for you to basically start feeling better. All right. So just to reiterate the fact that antigens are very specific to certain antibodies. Only certain antibodies on specific B cells will respond to those particular antigens. So you can see this particular antigen is only stimulating one particular kind of B cell. That B cell that is stimulated will clone itself, form plasma cells. Okay, you'll actually have memory cells formed. You'll have a huge number of these plasma cells that will form, and they will start creating a large number of these antibodies. Okay. Oops, I'm sorry. I tried to grab my face here. Let me unink this. So one last graphic, just to show you one more time. We have to have the B cell has to join with and display the antigen that it is specific for. So those antibodies on that B cell have to be bound to the bacterium or virus or whatever it's bound to. It has to display it on its MH2 protein complexes. Helper T cells also have to be activated by that specific antigen. So we have activated helper T cells will create those cytokines that will stimulate this B cell to form clones of itself, form plasma cells that will produce antibodies, and also form memory cells. Those antibodies, we need to talk about what those things do. So you can see that if you're exposed at time 0, it takes almost 7 days for those plasma cells to differentiate and begin to produce antibodies. We get pretty good antibody production at about day 14. So, in between day 7 and day 14, you start feeling better each day because you're binding up toxins or you're destroying organisms that are causing you to be ill. Okay? Now, a lot of us wait till about day 7 to go see a doctor. Maybe you don't wait that long, but usually we wait till about day 7. And then we go to the doctor, and all of a sudden we start feeling better. And it wasn't really necessarily something the doctor did. It's because our own immune system started working and we start feeling better. So those antibodies are produced after exposure. Those antibodies are produced, for several weeks. And then they decline in number. You don't need to keep on making that antibody after you've completely ridded your body of that particular organism. But if you are exposed to that organism at another later time, you already have memory cells ready to respond. So those memory cells will clone themselves really quickly, become plasma cells, and you have a no delay response at producing, logarithmic levels of antibodies, huge quantities. And they go up really, really rapidly, and they stay elevated for long periods of time. so that if you're re-exposed to that particular antigen, you continue to destroy that antigen. Now, the depressing thing about this is that if you're exposed to a different antigen, it takes the same level of time so that it took to the first antigen. So every time we're exposed to a new antigen, it takes a period of time for us to develop immunity to it. So if you get a vaccine, it takes time for you to develop antibodies to it. Okay, so if you take and you get the flu and then you get the vaccine, it takes time for antibodies to respond. So you want to get the flu vaccine right before flu season so that you'll be ready, so that when you're exposed to the flu, boom, you're producing huge numbers of antibodies and you don't get sick from that particular organism. Okay, so vaccines are real important because you have memory cells that will remember and have a no delay response next time you're exposed to that particular antigen. So just showing you again that it takes about 2 weeks for you to get that peak response by antibody production. Okay, so and they won't, these antibodies won't remain elevated for long periods of time. Next time you're exposed, though, those antibodies will surge and be significant. I kind of like this graphic because it kind of indicates to you when you're exposed to some organism that's trying to eat you for food, kind of how the responses are working. Your neutrophils are working real quickly, and then they wane off after about 2 weeks. We got natural killer cells; those things are working real quickly. Macrophages, those things are working pretty quickly. They are pretty long active, for a week and a half or two. But any of our innate, our acquired immunity takes a little time for that stuff to get stimulated. The cytotoxic T cells take a little bit of time. Plasma cells take a little bit of time, and antibodies, those things take time to get going, but they do last a little bit longer. Okay, so we have a lot of ways to kill an organism that comes in. Hopefully, one of these ways will work, because if they don't work, then we may die from the exposure to those organisms. So humoral immunity comes in two flavors, an active and a passive. Active humoral immunity is naturally acquired. It's where you actually acquire humoral immunity with an active infection. This is the best type of immunity that you can have. It seems like when you have had an actual infection, it stimulates the immune system really strongly, and your body really remembers when you have an actual infection. Artificially acquired active immunity is in the form of vaccines. Vaccines aren't as good as naturally acquired immunity, but I don't want to acquire immunity to polio naturally. I would much prefer to get an artificial vaccination as opposed to be exposed to the real organism and have to acquire immunity to it naturally. Okay, so vaccines are a good line of defense. We're killing we're having the ability now to put vaccines in people that help to protect us against cancer. When you look at the HPV vaccine and hepatitis B vaccine, these are actually cancer vaccines. But we also have viral and bacterial vaccines we can take as well, so that we can prevent ourselves from getting that. We also have gene guns and edible vaccines. There's all different kinds of ways other than shots that we can get vaccines. So passive humoral immunity includes natural means. So there's natural passive, which are antibodies in milk. Those are antibodies that you're getting naturally from your mother when you drink breast milk. We then have artificial passive immunity. This is where we take serum, so the plasma out of people's blood who have been exposed to an agent, and we can use their serum as and inject that into you, and those antibodies from that person that's immune to that or is producing antibodies to that antigen will now give you immunity to it as well. Antivenom is where we harvest the plasma from animals that have been injected with various kinds of antigens, and we harvest their plasma and harvest the antibodies from their plasma, and then we inject that into people, and that's how antivenoms are created. Antitoxins are the same kind of way. So we use the antibodies produced by humans in serum or animals in antivenoms and antitoxins, and that's what we use to treat things like rabies or the serum we might use to treat some kind of poisoning that you might have, or an envenomation. So antivenoms are an artificial passive form of humoral immunity. So antibodies are also known as immunoglobulins, and they're secreted by plasma cells, which are activated B cells. They bind to very specific antigens, and they consist of 4 polypeptide chains that are held together by disulfide bridges or bonds. There's 2 heavy chains and 2 light chains, and this is what they look like. So this is a structure of an immunoglobulin. We do have different kinds of immunoglobulins. This happens to be immunoglobulin G. So we can see that there are 2 light chains here, and there are 2 heavy chains here and here. And light chain means that it's just smaller, and the heavy chain means it's bigger. So there are these regions here that are variable. These bind to very, very, very specific antigens. And you make all different kinds of variable regions so that you can fight literally thousands and thousands and thousands of different kinds of antigens. So we are genetically programmed to make certain variable regions. So here's your disulfide bond or bridges that are holding these chains together. So this is a pretty complex protein having quaternary structure. Has basically 4 polypeptides that are held together. There are sites for attachment for phagocytic cells. So if this antibody sticks to a bacterium, there's a place here it's almost like a bullseye that tells a phagocytic cell, hey, here's where you need to target. Complement will also stick to this antibody and become activated. So there are regions for that to occur as well. So antibodies work in a variety of ways. They can agglutinate, that means stick to the surface of a toxin or a bacterium or a virus, and basically just stick around it. They can precipitate toxins that are water soluble. They can precipitate them and make them non-water soluble, so they'll basically come out of solution and then maybe not be toxic any longer. You can neutralize antigens like for example, you can neutralize venom components by sticking antibodies to those components and deactivating them. And then also antibodies will can activate those proteins made by the liver in the plasma of your blood called complement. And complement, when it sticks to an antibody, and becomes activated, can cause opsonization, so it can target a specific organism. Complement can cause chemotaxis, which is the movement of white blood cells out of your circulatory system and towards where a bacterial infection might be occurring. Complement can induce inflammation to occur and all the cool stuff that occurs with that. And of course, complement can also lyse any kind of target cell that it's joined on or that it's on. And just graphically shows how antibodies can work. So basically, we're having over here neutralization of a virus or a bacterium. So the binding of an antibody can also cause opsonization or targeting by phagocytes and phagocytosis. Agglutination is just binding it all up so it can't even move or have any activity that would be damaging to the body. Precipitation of soluble antigens, making them insoluble so they don't circulate very well in fluids anymore. Activation of complement. So an antibody right here is stuck to this foreign bacterium, and the complement proteins are activated, forming that complex that creates pores. So that attack complex of proteins that forms pores and destroys the bacterium. So here's the lysis of the bacterium when that pore forms. So all of these things, in terms of viral or bacterial neutralization, agglutination, and precipitation, these things can enhance phagocytosis and the destruction of a foreign agent. Now, there are different kinds of antibodies. The way that we remember the classes of antibodies is by thinking of the acronym MAG. So there are immunoglobulin M, immunoglobulin A, D, G, and E. Each of these has their own little specialties that they perform. So IgG is the largest and most diverse class of antibodies. It accounts for about 80% of the antibodies and they're responsible for resistance against viruses, bacterial toxins. If you remember back to the Rh factor, so the hemolytic disease of the newborn, where mom's antibodies cross the placenta and destroy the baby's body, IgG antibodies are responsible for that reaction. So IgG antibodies are anti-Rh antibodies. Okay, these IgG antibodies also give passive immunity to the fetus during embryonic development. Those ones that cross the placenta. All right, so IgE attaches specifically to the surface of basophils and mast cells, and when they're bound to that, they stimulate these basophils and mast cells to release histamine and other chemicals that can accelerate or cause inflammation. And if you have allergies, you can kind of blame IgE antibodies for that. So IgD antibodies, these are going to basically play a role in the sensitization of B cells. They basically will sit on the surface of B cells and activate B cells. IgM antibodies are going to be the first antibody that's oftentimes secreted when any antigen comes into the body and that we're exposed to. It's going to basically cause agglutination. Is one of the big things that it's going to form. It does circulate in a 5 antibody starburst, so it's very easy for it to stick to something and cause agglutination. And if we think of anti-A and anti-B antibodies, those are going to be IgM class antibodies. Okay, so if you produce those antibodies, those are IgM. So IgA is going to be found mainly in secretions that are glandular secretions. So we're talking about mucus, tears, saliva, semen. These are going to produce these are where IgA antibodies are going to be found. So before antibodies even come into your body, these particular antibodies are trying to prevent them from becoming internal. So let's see. So that's a little bit about the different class of antibodies. Just something about allergies. Sometimes we do have these things called allergens, which are proteins in the environment that aren't really deadly to us, but we do have, unfortunately, sometimes we do have B cells that recognize them. So you can see here this pollen grain. There is an antibody in this particular person that can be sensitized to that allergen. Plasma cell will form, Ig antibodies will form. Those Ig antibodies will be placed upon mast cells and basophils, and when the allergen comes into your body again, those antibodies that are stuck there are going to be stimulated to cause mast cells and basophils to release huge quantities of histamines, leukotrienes, prostaglandins. It's going to cause pain and inflammation, and it's going to cause all those signs and symptoms of allergies. So we do have the ability to utilize monoclonal antibodies. So we can actually take specific kinds of B cells, activate those B cells, and then harvest the antibodies that they produce. So if we want to use antibodies to detect things like HIV, we can actually create B cells that do that, harvest the antibodies, and then use it in a test for HIV. So you can go to the store and buy monoclonal antibody tests. So a pregnancy test is going to be based on using monoclonal antibodies to and it's going to be used to basically attach to human chorionic gonadotropin, which is a hormone released by a fetus. So if you have a fetus, if you have an embryo, it produces this hormone, and if you take and urinate onto one of these pregnancy tests, those monoclonal antibodies will actually tell you if you're pregnant or not. We also use monoclonal antibodies for blood typing. You did it in lab when we tested for our different blood types. So we had anti-A and anti-B and anti-RH antibodies. And then we can use antibodies to target cancer cells. So hopefully you're familiar with doing the blood test, because we did that in lab. This is how we make those monoclonal antibodies. We will take and challenge some kind of a model organism like a mouse or a rat. We will take and activate the immune system cells, the B cells. We will fuse the B cells with cancer cells, create hybridomas, and these will be cultured and then made to make antibodies in huge quantities. Okay, so this kind of just puts it all together, just to refresh your memory. When we're exposed to all these things that want to eat us, we have innate immunity and we have adaptive or acquired immunity. Innate immunity is going to utilize complement, all kinds of phagocytes, natural killer cells, macrophages, to try to kill anything entering into the body. So anything that makes it into the body that particular element of the immune system can't fight or destroy, then we're going to activate cell-mediated immunity and antibody-mediated immunity to utilize cytotoxic T cells and helper T cells and B cells in order to destroy anything that comes into the human body. Okay, so that's a little bit about the immune system that I wanted to cover with you. So make sure you do all your reading, study guides, do everything you need to do to be a great student. So I'll see you next time.